Hi, my name is SamuelWycoff, and this is the behavioral and physiological effects of nicotine on crayfish. Now, when people smoke, they just throw their cigarette butts onto the ground, like here. And the cigarette butts can get washed away by rain down these storm drains, and eventually they can end up in streams and rivers. And the organisms that inhabit these streams and rivers can suffer from the nicotine that's still present in the butts. So I designed an experiment around testing the full effects of these nicotine on those stream creatures, and I chose to work with crayfish, the species Procambarus clarkii, because they're pretty easy to work with, they're relatively easy to get, and also because they're also known as sentinels of water quality, so any stimulus that affects the crayfish is also likely to affect several other organisms in the ecosystem. To test the effects of nicotine on these crayfish, I divided my experiment into 4 separate parts. The first was behavior, which tested the crayfish ability to respond to its environment, which I defined as this tail flip behavior. When the crayfish get tapped on the tail, their tail curls underneath their body, which is a good indicator that the crayfish can sense that something just touched it. It's an escape response, and a good way of showing that the crayfish is indeed responsive to its environment. So what I did was I took 9 crayfish at a time, put them each in their separate tank filled with 800 milliliters of water. Some I left as control, some I made a high group, and some a low group. The high dose of nicotine got 15 milligrams per liter of nicotine, and the low dose got 7.5 milligrams per liter of nicotine. I got these values from a study done on another species of crayfish, which found that the LC50 for those crayfish was about 36 milligrams per liter. So I thought that a good high dose would be about half of that, and a good low dose would be about half of that. So after adding the nicotine to the crayfish in their tanks, I would take the glass stirring rod and I would tap them on their tails and record whether or not they tail flipped. And I did this every 24 hours since adding the nicotine to the nicotine groups. And I did this test for a maximum of 2 weeks. And what I found was the control, all of the crayfish responded for the entire duration of the test. 2 of the crayfish, one in the low and one in the high, died within 24 hours. The other 2 in the high group became paralyzed, and then the rest of the low and all the control were fine for the entire 2 weeks. Next, for my next trial, all of the crayfish all of the time responded to the tail flip. They tail flipped every single time, even the ones in the high group for the entire 2 weeks. Next, I tried to see the effects of nicotine on crayfish physiology and their internal bodily functions, which I decided to use heart rates and ventilatory rates, their breathing rates, essentially. To do this, I had to drill 2 holes in the crayfish for each measurement. 2 at the end of the gill chamber to measure the breathing rate, and 2 near the cardiac chamber to measure heart rate. And in these holes, I placed stainless steel wires, which were hooked up to impedance detectors, hooked up to a lab chart, which allowed me to record their heart rates and ventilatory rates. That's the mass movement of fluid as impedance, which then was converted into an electrical signal, which I could then analyze. Here it is on an actual crayfish. The arrows are pointing to where the holes are in the crayfish. These are what my recordings look like. Each little spike up here is a heart rate. Each of these blue spikes, the large ones, is a ventilatory beat. So this is the crayfish gill chamber in action. This is the cardiac chamber in action. And so what I would do for this test is I got 1 crayfish in its own separate tank, 800 ml of water, randomly assigned it as control, low, or high dose, and added the corresponding amount of nicotine. And then while I turned on the impedance detectors, which started this recording, and I would record for about 10 minutes, and then I would shake the tank just to startle the crayfish, and then record for 10 more minutes. To analyze this was the software I was using to record the data on, Chart 7, allowing me to convert the beats into rates. And how I analyzed this was I took the 1 minute before the tap and the 1 minute after the tap and found the percent change. Here's my results. For the control group without any nicotine, the heart rate didn't increase by or decrease pretty much at all. The ventilatory rate decreased by 11% though. With the low group, the 7.5 milligrams per liter of nicotine, the heart rate decreased by 26%, and the ventilatory rate decreased by 33%. And finally, with the high dose, the heart rate decreased by 42.5%, and the VR decreased by 47%. And then I ran an ANOVA test, an analysis of variance test, and found that there was significant differences between the heart rates, between the control low and high heart rates, but there weren't between the ventilatory rates. The p-value is about 0.1. So there's definitely a change in their heart rates, but not in ventilatory rates. So then I went back to the behavioral test and saw the paralysis and thought I could do something kind of with that, see the nicotine, probably the mechanism of that paralysis. What I did was, to understand the whole animal behavior in response to this, I used a neural circuit within the crayfish to see the effects of nicotine on it. And this is the neural circuit. Pretty simple one here. This is a section of the crayfish tail. There's a sensory nerve root processing the CNS, the central nervous system, and an output through the motor nerve. And what I did was I sacrificed a crayfish and dissected it, and then separated its abdomen, and then placed it in a special gel. And then I took this third nerve motor root and I sucked it into a suction electrode, which allowed me to record the nerve signals passing through that nerve. Here's what it looks like on an actual crayfish. It's kind of hard to see. This is the ventral nerve cord. This is the side going to the tail. This is going near the head. And this nerve right here, the third nerve root, that's where I hooked up the suction electrode to, which is where I was recording from, in an apparatus which looks like this. Imagine the picture you just saw was this little part zoomed in, a very large amount. So what I did was I placed the crayfish abdomen in this Sligard gel, or agar. I placed in it a modified Van Herveld solution, which just allowed the current in the nerve to flow. And what I did was, while recording the signals passing through that nerve, I took this paintbrush and I stimulated this. I brushed it, which would send a sensory input, get processed CNS out the motor nerve, which is where I was recording from. So I did 10 brushes at a time, and then I replaced the saline here with a saline mixed with the high dose of nicotine. And then I did 10 more brushes. And the results I got looked like this. These are extracellular recordings when each spike is an action potential. And as you see after this dotted line in the middle here, after the brush there was generally a large increase in the frequency of action potentials. And how I analyzed this was I took the number of action potentials 1 second before the brush and the 1 second after the brush and found the percent change in the two. But something that I noticed was after about 5 minutes of nicotine being in the solution, this happened. Suddenly the screen that I saw what I was recording from became completely saturated with these action potentials, with these spikes. And so I saw this was a large change from previous experiments, so I decided to dub this phase the late nicotine phase, and I did 10 more brushes in this late nicotine phase. And here's what I found. I found that on average, when the control, there was a 26.5% increase in action potentials, and then in the acute, just after adding the nicotine, there was a 26.8%. So no difference between those two. And during that late phase, though, that you just saw, there was only a 3% increase, which is essentially no increase. And so I ran an ANOVA test for this one, and there was not a significant difference between the three groups. However, there was definitely a significant change between the control and that late nicotine phase. The final part of my experiment was a habituation trial, which came across because I noticed two things. Two ways to tell if the crayfish were responding to a stimulus. One was these tail flips, which I did previously, and another was in heart rates. For when we scared and startled the crayfish, their heart rate paused for a second. Excuse me. The time between the pause greatly increased after the tap. And so these two phenomena, the tail flip and the heart rate pauses, were two ways I showed that the crayfish were responding. So what I did was I hooked up a crayfish similar to the physiology. I drilled 2 holes in the carapace, and piece detector's lab chart computer, but only for heart rate. You'll notice the crayfish has a piece of tape right here on their head. That's just to prevent the crayfish from noticing us before an actual tap occurs, to reduce some of the time errors there. And what I would do was I would record, while recording the crayfish heart rate, I would take a glass stirring rod and tap the crayfish on their tails every 30 seconds. And this would go on until the tail flips had habituated, which I defined as the crayfish wouldn't tail flip 6 times in a row. Which you can see here. This is one of the trials that I did. And at the beginning, the crayfish tail flipped, and it kept fluctuating until finally there were 6 instances in a row where it didn't tail flip. And I would repeat these trials, some with low concentration of nicotine and some with high in them. And again, I recorded whether or not they tail flipped, and I also recorded their heart rate pause length. So here it starts with pauses of 4-5 second gaps between each successive heart rate until it gets down to about just 1 second here. And as you can see here, the trial went on for about 1,400 seconds. However, the heart rate pauses seemed to stop changing really after about 600 seconds. So this is an interesting thing that I saw. So here's what I can learn from all of this. Because of the behavior results, only the nicotine group had anything happen to them. They died or became paralyzed. There may be a correlation between nicotine concentration and inhibition neurological function. However, it's not a very strong correlation, or it's just one that my sample size isn't large enough to prove. With the physiological test, nicotine definitely decreased the heart rate and ventilatory rates. In the CNS test, acute exposure to nicotine didn't do anything. However, a prolonged exposure caused great inhibition of the central nervous system. And the habituation trials are still underway, but so far it seems that nicotine is increasing the length it takes for her behavior to habituate, but I can't say so for sure yet. And I can take this a little bit further by changing my independent variable. In this test, it was just nicotine concentration, but I can do this test again with crayfish of different genders, ages, and masses and see how those differences affect how the crayfish reacts to nicotine. Something else I could do, I recorded from an efferent nerve and output nerve, but I also record from that first nerve, the second nerve, the input sensory nerve, and see if nicotine has different effects on sensory than output. And then finally, under periods of stress, crayfish are known to synchronize their heart rate and ventilatory rates, meaning that their heart and their gills beat in unison. So I can see the effects nicotine has on the synchronization. And then this can provide a gateway to see how nicotine affects other parts of the autonomic nervous system, the automatic one, the one that the crayfish can't control. And here are my sources, and thank you for your time.